Hello everyone, welcome back to the week 7 question answer session of the NPTEL course Experimental Biochemistry, which is conducted by Professor Shagota Dasgupta and Professor Shomode from IIT Kharagpur. I am Bias Mukherjee, a research scholar in Dr. Shomode's lab, and today I will be discussing with you the topics of week 7. And we will be solving a few MCQs and MSQ questions. Okay, so for today's topic we will be looking at the purification, so overexpression and purification of recombinant proteins, and what are the different methods, different biophysical techniques that are used for it. And this is something that we do in the lab on a regular basis, and so I can tell you the different machines, the different instruments that we use, and the principle behind every single step in this process. Okay, so let us start with the presentation. Okay, so first and foremost, why do we need pure proteins? So basically, pure proteins find application in the field of biotechnology, medicine, drug design, vaccine development, and also proteomics based studies. So very soon in the next slide we will look at all the different applications that pure proteins have. Okay, and so therefore to achieve all of these different applications, it is important to isolate and purify proteins. And so for the isolation, what exactly do you mean by isolation? So isolation is basically separation of a specific desired protein from other cellular components to obtain it in a pure and concentrated form. Okay, so this process is crucial for studying the structure of the protein, the functional assays that we perform using this, and also different applications. It finds different applications in biotechnology, medicine, drug design, vaccine development, and so on. So let us look at this image. This is basically the very concentrated form of protein that we are collecting, and this is possibly grown from a culture of around 1 liter, and this is in some micromoles. Okay, so the concentration of the protein is probably 500 to 600 micromolar. So then you understand the struggle. Okay, this is the small amount of protein that we get from the huge amount of bacterial culture. So basically, isolation and purification of proteins can be performed from natural sources also, alright, but then natural sources do not give you proteins in very large amount. So the proteins from natural sources that we obtain, they are very little in quantity, plus it is very difficult to isolate and purify those proteins, and therefore to maximize the yield, what we do is we create recombinant proteins. So these recombinant proteins are genetically engineered, okay, so and they are prepared in host organisms. So again, the host organism can be a bacteria, it can be yeast, it can be insect cells, plant cells, mammalian cells, based on your requirement and also based on the requirement of the protein. Okay, so for example, the protein needs some kind of post-translational modification, let's say glycosylation. Then it is very difficult to do that in bacteria, right? So then we will have to maybe think of a different strain of bacteria or some other alternative host organism. So this is how, based on the requirement of the protein and the requirement of the downstream studies, different host organisms are selected. And the recombinant DNA technology allows the insertion of the gene encoding specific proteins with desired functions in the host organism. So this is cultured, this host organism is basically cultured to produce the protein in large scale, and the biophysical techniques are employed for the extraction and purification of that desired protein. Alright, so now the question is, we understand that this is a very long procedure and we need a lot of different biochemical processes and a lot of heavy instrumentation for this, but then what is the need for pure proteins? So in this slide we will look at the different fields in which pure proteins find application. Okay, so protein isolation and purification are crucial steps in various scientific and industrial processes to obtain high quality proteins for a wide range of applications. The high purity of the isolated proteins is essential for reliability and accuracy of the different functions that the proteins are made to undergo. Hello, welcome back. We are looking at the isolation and purification of proteins today. So let us carry on with the presentation. You tell me if you are able to see it. Okay, so we will look at the different fields in which pure proteins find applications. Okay, so they are biomedical research, where the function of the protein is studied, structure and its different implications in diseases. So that is studied. Then there is drug development. So in drug development, we study the drug screening, validation, right? So maybe this protein is a drug, or maybe the drug is a small molecule which is binding to a protein receptor. So it can be either way, and we need these proteins in pure forms to study all this. All right. Then there is biotechnology, where maybe the therapeutic proteins and enzymes are being produced. Then clinical diagnostics. Okay, so development of diagnostic tests using specific proteins. So the protein can be a biosensor, it can be a biomarker. Again, in vaccine development, there is antibody engineering and there is protein engineering. So then enzyme catalysis studies, again there is enzyme engineering, metabolic engineering, all of these things go on. Investigation of the enzyme kinetics and mechanisms, and then also in structural biology. So I am in a structural biology lab, and X-ray crystallography and NMR studies are done. That is basically studying the structure of the protein. Also your cryo-EM comes in. Then there is proteomic studies. This is done to understand the interaction between different proteins, and also let's say in a particular disease condition, what all proteins they are getting, what all genes are getting upregulated, and what all proteins are getting produced in more number, and what all proteins are the production is getting reduced. So that can give you an idea about the gene, about the disease condition. So all of these omics studies go on. Then there are functional assays, so that we can try to understand the function of different proteins in a biological system, and also biopharmaceuticals, where therapeutic proteins are produced in large quantity. Okay, so this tells us that pure proteins and in high concentration is very, very essential in all of these different fields. And now let us try to understand how these pure proteins are obtained. So you can look at this slide. Here they are telling you that proteins can be extracted from natural sources as well. So see, there are different natural products that can be grains, that can be seeds, it can be your pea is there, then different flowers, leaf extract, all of these different different natural sources can be there, and from there different procedures are performed. So this is called pre-processing, where it is soaked and it is dried and it is grinded. And then what happens, there is wet extraction, where you have cell disruption procedures. Then the protein is extracted using alkaline or acid extraction, then salt extraction, and all of these different kinds of methods are used for the extraction of the protein. Then it is basically concentrated. So see, isoelectric focusing and all of this is done, and then the protein is basically exchanged into a different buffer, so using dialysis, and then the protein is kept in a suitable buffer. And then what happens, again it is heat treatment is performed and the protein is concentrated or isolated. There can be dry extraction, where different physical methods are carried out, and then there is electrostatic separation. So you can go through this slide, but we will not look at it in details, because we do not have any questions related to extraction of proteins from natural sources today, alright. But then all of these images have been taken from research articles and research papers, so you can do something. So review papers and research articles, so I have provided the link at the end of the PPT. You can always access that and you can look up this particular paper, alright, and you'll get a better understanding of this entire concept if you're interested. right? So now we will look at the overexpression and purification of recombinant proteins. So this is what we are doing in the lab on a day-to-day basis. Okay, so let us try to understand what goes on here. So what do you mean by overexpression, and what is meant by recombinant proteins? So recombinant proteins are basically non-natural proteins. So what does that mean? Here what happens, we take a host organism. Inside the host organism there is a, see, this is the gene. This gene is the one that encodes this particular protein. Now we can even make modifications in the gene, okay, so that a new variant of protein gets formed, and that is functionally more useful and it is more stable. That is called protein engineering. So our lab works with protein engineering and structural biology. Okay, so what happens, we insert this particular gene that is coding for this particular desired protein into a DNA vector. So what is a vector and how is it different from a plasmid? So a plasmid is a double-stranded circular DNA and it is natural, okay, but a vector is one which is modified. So that means it has been modified using genetic engineering, and certain sequences are added into it, certain unnecessary sequences are removed from it, DNA sequences, and that is how we get a expression vector. Okay, so here they have actually made it synonymous, this expression vector and expression plasmid, they have made it sound synonymous, but okay, so the connection is lost again. Let's see. Yes, so we were looking at the overexpression and purification of recombinant proteins. So what happens, basically this particular gene coding for a definite, a specific protein sequence, that is inserted into this vector. Alright, so you've understood the difference between vector and a plasmid, right? So now what happens, this is a expression vector. Expression vector means that it is going to have this ribosome binding site and it is suitable for translation. Okay, so ribosomes are going to come and sit onto this vector and it is going to code for the specific protein that we want. Then what happens, we go for transformation. So what is transformation? Transformation is basically making your plasmid enter into a competent bacterial cell. So competent cell is a cell that is having the capacity to take up foreign DNA. Okay, and how do we make bacterial cells competent? We give a heat shock and we use CaCl2 solution for making the bacterial cell competent. So you can look up at the different proteins that are necessary for rendering competence in bacteria. So all of that you can have a look, but right now we will be talking on the overall process of overexpression and purification of recombinant proteins. So I am sure you understood what recombinant protein is. So recombinant, it is being called recombinant because, okay, 1 minute, yeah, so it is being called recombinant because it is getting inserted into a plasmid DNA and this is not natural, right? That is why it is genetically engineered. That is why it is recombinant. Second thing, what is overexpression? So this bacterial culture is getting formed. Okay, so basically we take a 500 ml or 1 liter or even more volume of bacterial culture and then we grow it at, let's say, 37 degrees centigrade, and then what happened? we induce this protein expression. So for that we add a chemical called IPTG, which is, we'll learn about it very soon. So this is added, and when this is added, protein synthesis begins. Okay, so we will look at the entire genetic makeup of the bacteria, why IPTG is inducing protein expression. We will have a look at all of that. Okay, so basically inside the bacteria your protein is getting formed. Then what happens? We are going to spin down all the bacteria. So entire media is then thrown away, and you take the bacterial pellet, all the bacterial cell go to the pellet. Okay, and this pellet, what you do is you add suitable buffers to it, and then you go for sonication and enzymatic degradation of the bacterial cell wall using lysozyme. So in sonication, there is ultrasonic wave which is passing through the bacteria. It is rupturing the walls of the bacteria. So all of the protein that is there inside the bacteria, that is coming up. Okay, now why is it called overexpression? So it is called overexpression because apart from the bacteria's own protein, we are adding this special DNA in the DNA vector. So inside that we are adding a viral polymerase gene. So what is it doing? So there is a T7 bacteriophage polymerase, which is very much efficient, more efficient compared to the bacterial DNA polymerase. And what is it doing? It is improving protein expression 5 to 10 times compared to the bacterial DNA polymerase. And that is why whatever expression of protein is normal, our protein is going to be formed a lot more than that, and that is why it is called overexpression. Right, so all of this has been genetically engineered, and that is why we are overexpressing our desired protein in bacteria. Right, so then after that what happens? We go for sonication, and then we again spin down everything. So all the cell debris, so the cell has been broken, right? So all the broken bits of the cell, all of that, we centrifuge it, it comes down at the pellet, and then we collect the supernatant. So supernatant is going to contain your soluble proteins. So we take the supernatant, we add PMSF, and PMSF is a protein serine inhibitor. So we will, protease inhibitor it is basically, that means it is going to stop all the proteases from degrading the proteins. Okay, all the bacterial protease enzymes, all of that is also coming out from the cell, because the cell, the different compartments of the cell are basically all broken, right? So these protease enzymes should not be harming your protein solution, desired protein. That is why we are adding PMSF. Then what happens is, then we take that protein for purification, and we have studied the different chromatographic techniques. So we know that there is ion exchange chromatography, there is NINTA, histag, right, affinity chromatography, and then see, this is the column. You can look at the column here. So we are going for chromatography, you can see the column, and then after that we are going for protein structure and functional analysis. So this much I am sure you have understood. Fine. So we will be looking at the next part. So see here, what are the advantages of overexpression and purification of recombinant proteins? So this entire method gives you a very high yield of the desired protein. Next thing is customization. So this allows expression of the protein with specific modification that I was telling you. Genetic engineering, protein engineering, right? So you can make modifications at the nucleotide level, and then you can have different tags. So these are different affinity tags. There can be GST tag, there can be his tag, there can be flag tag, right? So all of these different extra proteins you can add before your protein to improve the solubility of the protein. For example, you are adding ubiquitin, you are adding RFP, right? So all of this you can do. So different customization can be performed. Then cost effectiveness. Of course, this is a cheaper and a faster method compared to isolation of protein from natural sources. Then comes in reproducibility, right? So when you do the same experiment again and again, and if you keep all the conditions exactly the same, it is expected that you are going to get exactly the same result. All right. Then there is purity. So you get very pure protein compared to what you would otherwise get from natural sources. Again, the speed and efficiency is a lot better. Okay. Now let us look at whatever I told you. Let us look at that in this image form. Okay, so before this we understood that there was this gene, it was getting integrated into your plasmid, right? And then it was forming this double-stranded plasmid, and this is getting transfected inside the bacteria. And what happens inside the bacterial culture and what happens after that? So let us look at that here. See, expression in E. coli. So what happens? Inoculate the media. What is inoculation? Inoculation is nothing but adding of the bacterial culture into your let's say a culture media. And here we often go for LB media, which is Luria Bertini broth. We also go for minimal media. Okay, so there can be different kind of media based on your design, preferences, right? So what happens? See, here what we are doing is we are basically adding this. So this is called inoculation. Inoculation means adding the bacterial culture in the media. Then what happens? Induce protein expression. So induction is done using IPTG. We will look at IPTG and what is the chemical composition and what does it do very soon. Okay, so just try to understand that IPTG is a chemical when you add, the lac operon inside the bacteria gets activated, and then what happens? Just next to the lac promoter there is a gene for RNA polymerase sitting. So that gene gets activated and T7 RNA polymerase, viral T7 RNA polymerase gets produced. This T7 RNA polymerase goes and activates the gene for your desired protein, and it starts getting expressed. Then the protein starts getting formed. Okay. Then there is harvest. So what is harvestation? This is nothing but getting the entire bacterial cells down in the form of a pellet after centrifugation and getting all the culture in the soluble fraction. Then what happens? So after that, okay, so in between there is a step, there is sonication. Okay, so the cell lysate is prepared, and then what happens? We go for sonication. So if our protein is coming in the pellet, then we have to go for sonication, and then we have to spin it down again, and all the cell debris is thrown and all the supernatant containing the soluble protein is taken. Now that is filtered. Okay, that is syringe filtered through a filter of 0.22 micrometer, and then it is made to pass through this NINTA resin. Then what happens? First the flow through is collected. Flow through is containing all the bacterial proteins, and our own protein is basically bound to this column, because our own protein, it is genetically engineered, it is recombinant. That means it is containing the histidine tag, hexahistidine tag. All the histidines are going to bind to your nickel in the NINTA column. This is nothing but affinity chromatography. So then what happens? The protein remains, your desired protein remains bound, and all in the flow through all the other bacterial proteins pass, right? Then what what do? We increase the imidazole concentration in the buffer. What does imidazole do? It comes and displaces your protein. It gets bound to nickel NTA. So all the protein comes in as elution, right? So pass elution buffer with 250 millimolar imidazole. So what we do is we use 500 millimolar imidazole so that all the bound protein gets displaced. All right. Then what we do is we go for this dialysis and histag removal. Okay, so this is a dialysis bag which is having a semi-permeable membrane, and what happens here is that through the semi-permeable membrane all the buffer components are passing and your protein is there because the protein is heavier. So semi-permeable membrane, it is having small pores. Proteins are too large to go across the pores, so it is all there. So basically what happens, initially the protein was in a buffer with a lot of imidazole, but imidazole is not very good for protein activity. So what happens, we exchange the buffer, we change it to, let's say, phosphate buffer or any other buffer in which your protein is going to be more comfortable, right? Then what happens after dialysis? See, we concentrate the protein, and then also we can go for size exclusion chromatography, and we can check whether the protein that we have received, that is pure or not, and also what is the size of the protein. So there is also one more step they have added, which is adding the protease to the protein, which is histag removal. So after the histag there is often a small protease cut site. Okay, in our case it is thrombin, it is TEF protease cleavage site. So what happens, you want to remove the histag and then you want to go for other studies, okay, functional assay or any structural assay of the protein. So for that we introduce this protease site. So what happens, we add this specific protease enzyme and it goes and cleaves there. So what happens, histag is like 2 kDa and your rest of the protein is there, and then you go for size exclusion chromatography. So your protein elutes separately, and then when you concentrate it, since it is 2 kDa, just 2 kDa, and the concentrator, this is called centricone. So the cutoff for the centricone is 3 kDa, right? So your protein remains and the cut portion of the protein goes out. So basically it is removed, and then you go for ACC and you get the pure protein chromatogram. Then you go for SDS page. Here you can confirm the purity of the protein and also the concentration of the protein. Look at the intensity of the band, look at no other bands in that same lane. So that tells you that the protein is pure and the protein is highly concentrated. So this is the entire process of overexpression and purification of recombinant proteins. Okay, now let us look at the different steps in more details. Again, okay, someone is ready to join. Okay, so we'll carry on with the PPT. So today we are looking at isolation and purification of recombinant proteins. Okay, now we will look at the different steps in greater details. So have a look at this. Yeah, so there are these different steps. So the first 3 steps that you see here, these are basically gene cloning. So what is happening here? As I told you, the desired gene is there. That desired gene you are integrating into a vector. Now when you are creating this desired gene, you need to keep in mind the codon optimization. So what is basically codon optimization? This is the fact that there are different tRNAs inside the bacterial cell, right? So for each codon there is only one tRNA and the number of that particular tRNA varies. Okay, so let's say the codon for, let's say, glycine, it is GGC. And there is another codon for glycine, since codons are degenerate. So more than one codon can code for the same amino acid, right? So another codon, let's say GGU. But what happens for GGC, let's say there are 3 amino acids, and let's say for GGC there are 2, no, not amino acid, there are 3 tRNAs that are going to bring that amino acid corresponding to that codon during the translation step. Okay, but for GGU, let's say there are 2 tRNA. Okay, that is called codon bias. That means what sequence should be there for each codon in my gene sequence such that I get more number of tRNA to bring that specific amino acid for protein expression. So that you need to find out, and for that the resources are available online, where if you search codon bias in bacteria, you get for which codon what is the percentage of tRNA present in bacteria, and based on that only you need to synthesize your gene. Okay, now what happens is, so that is one thing. Then we will look at the next step, which is cloning, and then we will transform it into the competent bacterial cell, and then we will grow it. Okay, from a plate of bacteria we are going to select a colony, we are going to know the gene sequence, we are going to go for DNA sequencing. Okay, so after DNA sequencing we are going to know what is the sequence of the gene inside that specific plasmid. We are going to isolate the plasmid and we are going to send the plasmids for sequencing. After that, if the sequence matches with your desired gene, then what happens? Then we will take that bacteria, we will culture it in a larger growth media, let's say LB media, Luria Bartini broth, or minimal media, anything that you prefer. Then we will go for the entire isolation purification procedure that we just talked about. Then we will target protein, we will collect the target protein. Okay, so once again, going through all the different steps, there is gene cloning, which involves the isolation and amplification of the desired gene. This gene is then inserted into a plasmid. See, this is the one, which is a small piece of circular DNA that can be easily replicated and transferred into the host organism. So then the host organism is transformed with the plasmid using techniques such as electroporation or chemical transformation. Okay, this allows the host organism to express the protein encoded by the inserted gene. Expression of the protein involves inducing the host organism to produce the protein. So induction, yes, we will learn about induction very soon. So for induction we are going to add a specific chemical which is going to make the host organism make this protein. Okay, this can be done by adding specific growth conditions or by adding a chemical inducer, which is often IPTG, right? Then the protein is harvested from the host organism, usually through centrifugation or filtration, and the purification of the protein is a vital step in the process. It involves removing any unwanted, so purification basically means separating your desired protein from any other unwanted contaminants, okay, which can be accomplished through various techniques such as chromatography, precipitation and filtration. Okay, now we will look at cloning and expression of the desired protein. So you need to understand two things first. One is what is a cloning vector and what is an expression vector. So we know that vector is a genetically modified plasmid. Okay, what is the difference between a cloning and an expression vector? So cloning vector contains your gene of interest and it contains the ampicillin resistance or any antibiotic resistance site, so that it can be selected against all other different bacteria when we are growing it in a plate containing that specific antibiotic. So this is basically a selection pressure. Third thing that it is having is this ORI site, which is independent replication site. Fine. And then there is this multiple cloning site into which you insert your gene of interest. What about an expression vector? So expression vector is designed for protein expression. So it should have a start codon for starting of the protein translation. It should have a stop codon for ending the protein translation. It should have ribosome binding site before the start codon, where ribosomes are going to come and bind and then start the entire protein synthesis, right? So this is the ribosome binding site, and then it is obviously also going to have a selectable marker, antibiotic resistance, and it is going to have this gene encoding repressor that binds to the operator and regulates the promoter. So this entire lac operon system should also be there. This is an inducible operon, right? So inducible promoter system should also be there. Now we will have a look at Now in our cell, actually this inducible system, this is integrated into the genome of the bacteria. We will soon look at it, but the basic difference between this expression and cloning vector is that expression vector should have start codon, stop codon and ribosome binding site. Most important 3 things. See, promoters, enhancers, terminator sequences, initiation sites, etc. Okay, now how to find out what kind of cell I should use for my desired protein. So see, there are HEC293 kidney cells, human kidney cells, there are CHO cells. These are all mammalian cells. This is needed for post-translationally modified proteins. Okay, but then these are, again, the culture duration is longer and is very expensive. And then there are insect cells. Again, you can go for high density culture and post-translational modification is possible. But then again, partial glycosylation only happens. E. coli, the ones that we use in our lab, this is low cost and it is easy to scale up, right, rapid expression, but you cannot go for post-translational modification. If the protein is going into inclusion body, it is becoming insoluble, then that becomes a big problem. Again, the molecular weight of the protein is also a limitation. So, you cannot go for very big proteins using E. coli. And then there is yeast. Only some post-translational modifications are allowed in yeast cells. It is low cost and it is easy to scale up. So, there is this table. In this table, you can have a look and know what are the different kinds of host expression system and what are the advantages of them, what are the challenges and what are the common applications. So, I told you everything in a gist, right? So, you can have a look here. See, everything is given here. This is very useful. So, take a look at this whenever you get time. So, after this, we will, okay, and this is basically, this actually tells you about the different bacterial strains for overexpression. So, we will come back to this slide very soon. So, the one that we were talking about, see, BL21. BL21 is E. coli polymerase, but T7 polymerase is not there. This is used for the expression of non-toxic protein and it can be used for the expression of used for protein expression using plasmids such as PMAL. Okay, then there is BL21DE3. So BL21DE3 is the one that we usually use in the lab. It is having this T7 Phage RNA polymerase. Right, so if you want to look at this particular cell, this is a BL21DE3 expression system. So what happens here is, see, this is the genome of the bacteria. Okay, so this is it is its DNA, it's the genome of the bacteria. Inside the genome of the bacteria, one small portion has been genetically engineered. So what has been done? Look at this tiny part, this is called DE3. It has been taken from a phage. Inside that DE3 portion, there is this lac operon, promoter is there, lac operon, operator is there. Okay, and next to it, so all the structural genes of the lac operon have been removed, your X, Y and A, all that has been removed, and the gene that codes for T7 RNA polymerase has been put inside. So what happens? When you are adding this, all the time mainly your lac repressor goes and binds to the operator site. So, promoter is inactive and T7 gene is inactive. So, no T7 RNA polymerase gets synthesized. But what happens when you add IPTG? IPTG is basically a structural analog of allolactose. Right? So, you tell me if you are not able to hear or if there is any disconnection or anything, tell me, okay? Okay, so we were looking at this. Yes. So, this IPTG chemical that I was talking about all this while, this is a structural analog of allolactose, but it is not metabolizable by the bacteria. So, what happens? This is a structural analog, so it comes and what happens? This lac operon system gets activated, right? And the repressor cannot bind to it anymore. So, E. coli RNA polymerase comes and binds to this promoter, lac promoter. Then what happens? The downstream gene, which is your T7 gene, this gets expressed. So what happens? T7 RNA polymerase, which is a viral DNA polymerase, this gets synthesized, and what happens, it has an inside, this is your PET vector, this is the expression vector, right? So inside the PET vector, again there is this site for T7 polymerase. So see, again this is also having lac operator, it is also having your T7 promoter, this is all genetically engineered. So what happens when you add IPTG, again this lac repressor becomes inactive, so T7 RNA polymerase comes and sits on the T7 promoter, and what happens, the target gene, whatever is the protein that you want to form, right, that starts getting expressed and the protein starts getting formed. So this is what happens after IPTG induction inside this bacterial BL21 DE3. So DE3 is coming from this modification, and there is also one tiny difference. So what happens in BL21 DE3 P lyase S strain? So P lyase S is a small vector. What it does is it contains a T7 lysozyme. So this protein, what it does is it comes and binds to the T7 polymerase in case of leaky expression. So we know that lac promoter is having leaky expression, that means even when repressor is bound to it, very tiny amount of gene is getting expressed because lac promoter is a leaky promoter, right? So even if small amount of RNA polymerase, T7 RNA polymerase keeps getting expressed, that is basically inactivated by this T7 lysozyme. So this is the entire story of BL21 DE3 expression system. Okay. Now we will look at the different steps in protein purification and overexpression. So look at this, the first step we already looked at the diagrams, and now we will just go through the theory. So I have included the theory portion for you to remember everything. Okay, so see, gene cloning, insertion of the gene of interest into suitable plasmid vector under the control of a strong promoter. Then what happens is transformation. So transformation, transferring the recombinant plasmid into competent bacterial cell using a process called transformation. Then there is cultivation and induction. So grow the transformed bacterial culture into suitable growth media and induce the protein expression by adding an inducer. This IPTG, it is also called isopropyl beta thiogallactopyranoside, and this is also the inducer in this case, right? Similar to your lacoperon. And then what happens? Cell harvesting, which means spinning down the cells in a centrifuge, and after a specific incubation period of 5 hours or more, this is the amount of time it takes for the protein to get produced in large quantity, and then the protein of interest is expressed at high levels. So resuspend the pellets containing the bacterial cell in lysis or wash buffer with a protease inhibitor like PMSF and sodium azide to prevent microbial growth. Right, so this is the structure of IPTG. You can see that this is actually a structural homologue of allolactose. This extra group is added here, and IPTG is a molecular mimic of allolactose, non-metabolizable. It triggers transcription of the lacoperon, and it is therefore used to induce protein expression where the gene is under the control of the lac operator. Got it? And then there is PMSF, which is a serine protease inhibitor. So what does PMSF do? PMSF stops the proteases, serine proteases, to act on the proteins of our interest. Okay. Then we will look at the other steps of other steps in protein purification. Okay, overexpression and purification. Right. So there is cell disruption. For this we use sonication and we add lysozyme into the cells. So what happens? Lysozyme breaks up the cell wall, and during sonication, ultrasonic waves are made to disrupt the walls of the cells. Then what happens? All the contents of the cells come out and the protein comes out in the solution. You centrifuge it. So if the protein is soluble, it will remain in the supernatant and all the cell debris and everything goes down into the pellet. Then what you do is you collect the supernatant, you syringe filter it through a 0.22 micrometer filter. Then what you do is you go for purification, and if it is a histite protein, you purify it using affinity chromatography. We talked about affinity chromatography and the different forms of chromatography in our last lectures, right? So you can go for affinity chromatography, and if it is not having an affinity tag, then you can go for other different methods of chromatography separation. So we will remove this, no problem. Okay, right? So then comes in dialysis and concentration. So we will exchange the buffer in which the protein is present. So we understand that we just talked about it, that a lot of imidazole was there in the elution buffer. So imidazole is not very good for your protein activity, and that is why we want to remove all the imidazole content, extra imidazole content. For that, what we do is we dialyze. We use a dialysis membrane. The pores, it is a semi-permeable membrane, so the pores are not large enough for the protein to pass through, but they are large enough for the ions and the buffer components to pass through, right? So outside the dialysis membrane there is one buffer. Inside the dialysis membrane, the protein in your lysis or imidazole buffer is there, and then the buffer gets exchanged, and then what happens, we concentrate it by using a centricone, and we spin it again and again so that the buffer comes down and the protein gets concentrated. That is, in a small volume, more number of protein molecules are there. Okay, then we analyze the purity of the protein using SDS page, and also check its molecular weight using, let's say, size exclusion chromatography or SDS page. Then we store the protein by adding PMSF and protease inhibitor cocktail. Cocktail refers to a mixture of different protease inhibitors, and then finally we go for the structural and functional analysis. Fine. So these are the different techniques. You can go through the different techniques. You might be knowing most of them already. So what is salting out, and then what is solvent, organic solvent precipitation, isoelectric precipitation we studied in our older classes, right? Then there is centrifugation. So differential centrifugation refers to spinning at different speeds, different RPM, so that different components get separated at different time points. Okay, at first the larger components, they will get separated, and as you go on, keep increasing the RPM and also the duration of the centrifugation, what happens, even the smallest of the components like ribosome, so all of these small ribosome, then your peroxisome, all of these small components, also microsomes, all of these get separated last. So that is differential centrifugation. And then there is density gradient centrifugation. So what happens in density gradient centrifugation is, you might have heard about cesium chloride density gradient centrifugation. So what happens in that is, just a minute. Yes, so in cesium chloride density gradient centrifugation, this is also called isopycnic centrifugation. What happens, cesium chloride is added, and also there can be sucrose density gradient centrifugation. So different percentage of components are there. For example, this is the most dense one is at the bottom, and it is like a gradient, okay, a gradient of components. And then what happens, your protein actually gets settled at the place that matches with its molecular weight. So let's say it is very heavy, then it will come towards the bottom, but then let's say if it is very light, it will settle in the upper layers, and that you can collect. So that is density gradient centrifugation. Then I told you about dialysis. Yes, 1 minute, connection is lost. Right, so we were looking at the different techniques of protein separation. Yes, we were looking at the different techniques of protein separation. Okay, so then comes in dialysis and ultrafiltration, and then the different forms of chromatography that we discussed in the last to last class. Right, so what is salting in and salting out? So this is a very interesting concept. Have a look at this. Initially, what happens when there are very few salt molecules, the protein salt interaction is much less compared to the protein interaction. That is why what happens, there are aggregation. Then as you keep on increasing the salt concentration, what happens, the proteins are very well interacting with the salts, and this is also enabling the proteins to interact with the water molecules. That is why what happens, the proteins are very well soluble, right? But then when there is too much of salt, what happens, all the salt ions, they are now interacting with the water molecules and the proteins are left behind. All the hydrophobic regions of the protein interact with one another and there is aggregation. So this is called the solubilization saline, and this is precipitation saline, right? So this is the zone of solubilization and this is the zone of precipitation of the protein where a lot of aggregation happens, and this is a nightmare for protein biologists. So this is what you need to do whenever you are talking about proteins. The solution in which you store the protein should have ideal amount of salt concentration so that the protein is not collapsing. Okay. Now we will again have a glance at the different chromatographic techniques. So we had already studied about all of these. I hope the connection is better now. Okay, so we have already studied about all of this. Look at this. This is ion exchange chromatography, where the positively charged matrix stationary phase is there, and to this the negatively charged analyte is going to bind. It is exactly the opposite in a cation exchanger, where to the negatively charged matrix positively charged protein is going to bind, right? And then this is anion exchange chromatography. So initially all the positively charged protein come out, then what happens, all the neutral charged proteins come out, and only finally when you increase the salt concentration and decrease the electrostatic interaction between the positively charged matrix and the negatively charged analyte does the anions, the negatively charged analyte come out, right? So this is the entire story of IEX. Then we will look at NINTA. We had studied this as well. So in NINTA what happens? Nickel NTA chromatography. This is affinity chromatography, where to this agarose bead this chemical group called NTA is attached, nitrilo triacetic acid, and this is chelating nickel, and the histidine groups of the protein are having these imidazole side chains, and these imidazole side chains are again forming coordinate bond with nickel, and what happens, 6 different histidines are introduced into the beginning of the protein, and then what happens, the protein remains bound to the stationary phase of the column, and all the undesired proteins without this hexahistidine tag go away as flow through. So this entire concept is called immobilized metal affinity chromatography. It is a form of affinity chromatography, and what happens is FPLC, which is fast protein liquid chromatography, exploits this NINT affinity chromatographic technique to purify his 6 tagged proteins, and elution is achieved by analyte like competitor, which is imidazole, and non-specific interaction, which is increased salt or ion concentration. Fine. Now we will be looking at the entire whatever we studied today. This is just a summary of that. Okay, this slide is nothing but a summary of whatever we studied today, and whatever you see new here, that we will be learning in the next few classes. Fine. So see, this is protein production. How do we do that? For protein production, there is strain selection. Strain means what bacterial strain you need. So based on the need, you will select the host organism. Again, you will select exactly which strain of the host organism you want. Then there is codon optimization, exactly what gene sequence need to be inserted so that there are more number of tRNA bringing that specific amino acid corresponding to that particular codon inside the mRNA. So that is called codon optimization. Then there is fusion system. Fusion systems mean, I told you that to increase the solubility of a protein, you can maybe include a ubiquitin protein in front of your own protein. So that makes it a fusion protein, right? So that is a fusion system. You can also go for these GST tags and also other tags, okay, in front of the protein. Hexahistidine tag. Then there is co-expression. So co-expression means there is a GST tag for one protein, and to that protein another protein is very tightly binding. So both of these proteins are basically co-purified and co-expressed. Then there is mutagenesis. You can go for protein engineering, mutate it, and then you can go for genetic engineering, isotope labeling. So for example, you want to go for NMR spectroscopy, then you can include N15, right? N15 nitrogen 15 isotope instead of the nitrogen 14 non-NMR isotopes, right? Non-NMR active isotopes. You can also go for C13 isotopes. Then there is also radio labeling. So if you want to go for radioactive analysis techniques, then you can go for radio label isotopes, right? All of that you can do. Then also there is protein purification. So purification is done for soluble protein. All of the processes that I talked about today was for soluble protein, but it can also be done for insoluble protein, which is folded. Let's say it is going into inclusion body. The protein is insoluble, it is going into inclusion body. Then you can read up what happens in that case. So there is a lot of washing and refolding steps performed, okay. That is called IB wash, inclusion body wash. So all of that different procedures are followed. You can have a look at that if you're interested. Then there are different membrane proteins. So membrane proteins are very huge, right? So it is very difficult to actually clone and purify membrane proteins. So how is that done? That you can learn about. And then there is cleavage of the fusion moiety. So yes, there are two different fusion proteins. You cleave using a protease, then what happens? That protein is removed and you are left with your protein of interest, right? And then there is characterization. So characterization includes physiochemical characterization like mass spectrometry, which tells you about the mass of the protein, the different post-translational modifications on the protein. It tells you about the sequence of amino acids in the protein. Then there is physical homogeneity, which you can come to know using SDS page. Then association stoichiometry. You can use analytical ultracentrifuge, you can use size exclusion chromatography to know whether there are more than one different kinds of populations in the species. For example, there are dimers, oligomers, monomers, what kind of stoichiometry is there in the protein. Fine. There is conformational stability or not, which means whether the protein, let's say it is binding to DNA, whether there are only few conformations and it is very stably binding to DNA, or there are too many conformations and the protein is like fluctuating and all the different amino acids are assuming different rotamers and the binding with DNA is very, very weak. So then the dissociation constant is weak. So that is the conformational stability of the protein. Fine. Then there is functional characterization like biochemical, immunochemical, protein interactions. All of these are functional characterization, and the different structural characterizations include protein crystallization, X-ray analysis, NMR, electron microscopy, etc. So see, here two different proteins are interacting with each other. One of them is an SH3. It is interacting via actin loop, and this model has probably been made using X-ray crystallography or NMR, and then the size has been confirmed using this your SDS page, right? So all of these different experiments can be carried out. Now we will look at the different different experiments that are involved in the analysis of the proteins after they are purified. So this is X-ray diffraction and crystallography. What happens in this case? X-rays are there and then you get this electron density map. This is the electron density map. This is the diffraction pattern, and you study this to get the electron density map, and you fit that to get the model of the atom. And then there is CD spectroscopy, which tells you what is the secondary structures of the protein. So see, for alpha helix you get this kind of a curve, for beta sheet you get this kind of a curve, and for random coil you get this kind of a curve at specific points, and from the peaks you can say what kind of secondary structure is there in the protein. And also for NMR spectroscopy there is sample preparation, data collection. This is the NMR machine, and then you go for this HSQC. So resonance alignments, these are the different amino acids, and peaks are obtained for the different amino acids, and then the structural studies are performed, and you get the model, right? So using NMR you can know a lot of things like the stability of the protein, what kind of interactions are there between proteins, and then the structure of the protein. So there are different things that you can come to. Fine. So now we will go into the assignments. Okay, so you try solving and you tell me in the chat box. If I get disconnected I will rejoin, otherwise I will look at the chat box and we can try to find out the question. So yes, first question is this: which of the salts? Okay, so you are saying it is C. Okay, ammonium sulfate, right? So we will take a break for 2 minutes and then I will be getting back. So let's take a break till 7:10 and then we will carry on with the MCQ questions. Okay. Hello everyone. So let us carry on with the PPT. Yes. So the answer is definitely ammonium sulfate. Okay. It is not given here, but yes, the answer is ammonium sulfate. So they are asking you which salt is the best for salting out, and for salting out ammonium salts are the best. Amongst these two, ammonium sulfate is the one that is commonly used for salting out of proteins. Okay, now we will look at the next question. So you can give me the answers in the chat box. Okay, so in the first question, it is in a mixture of 4 proteins listed below, which one should elute third in a size exclusion or gel filtration chromatography. So A has molecular weight of 13 kDa, B has molecular weight 68 kDa, 68.5, C has molecular weight 450 kDa, right? And immunoglobulin, so you are saying that B is the right answer, right, for this one. Yes, you're very correct, because see, for a size exclusion chromatography, what happens is, okay, 1 second, pen, yes. So for size exclusion chromatography, you know that the biggest one elutes first, and then it is the medium one, and then it is the smallest one. So the peak size, this tells you amount of protein that is present, right? So this is the basically order of elution in a size exclusion chromatography. So for this third one, in this case there should be 4 peaks. So for the third one, you see this is the first one that is going to get eluted, this is the second one, and this should be the third one that is going to get eluted. What do you think about the next question? In a mixture of 3 proteins of sizes 70, 10 and 25 kDa, in which order do you think they are going to elute? Okay, so you are saying it is B, all of them would come together. Why? Yeah, right. So 70 and then 25 and then it is 10 kDa, right? So we will look at the answer. Yes, so it is A in this case and it is B in this case, right? Now, question on the chromatographic techniques. Which of the following techniques can be used to separate a protein that binds strongly to its substrate? So this is exploiting a biological interaction. And in the next question, see, gel filtration chromatography separates proteins based on what? Their surface charge, their size and shape, affinity to the column, and pI. So the questions are very simple in this case. Yes, so you are saying that this should be C and the next one should be B. Yes, you are very correct. Yes. So see, now in this tiny table you can see that the properties of proteins. So the properties of the proteins and the different techniques. So size exclusion chromatography depends upon the diameter of the molecule. For ion exchange chromatography, it is the electric charge of the molecule, right? Reverse phase chromatography depends upon the presence of hydrophobic domains. Hydrophobic interaction chromatography depends on, again, hydrophobic groups, and affinity chromatography depends on biospecific interaction. So if you keep this tiny table in mind, you will be able to solve most questions of chromatography very easily. So see, for gel filtration chromatography, the proteins are separated based on the size and shape. Okay, now there are 3 different questions in the next page. Let's have a look at them. So in the first question, they are asking you which of the following techniques can be used to study the secondary structure of a protein. We just looked at 3 different techniques. Yes, okay. Try the next two. To determine the size of a protein, which of the following techniques can be used? So one is size exclusion chromatography, another one is your SDS page, and then there is analytical ultracentrifuge, and D is all of the above. Yes, that is right. all of the above can be used for determination of the size of a protein. So which of the following techniques can be used to determine the three-dimensional structure of a protein? B and C. Very good. Okay, let's see. Yes, so all the answers are very correct. For studying the secondary structure of a protein, circular dichroism can be applied, and SEC, SDSPH, AUC, all of these can tell you about the size of a protein. And for the last one, you see DLS basically tells you about the size of a protein, but 3D structure of the protein can be given by NMR, cryo-EM, and circular dichroism tells you about the secondary structures of a protein. Okay, so now we will move on to the next two questions. Okay, this I think is the same. So we will just keep this. Yes. So we'll go to this tiny question here in order to lyse the cells. This is very easy. What kind of process breaks open? Yes, sonication. So it has to be related to sound. We use ultrasonic waves. So it is definitely sound energy. Then imidazole can bind to. To whom can imidazole bind? It is A. Okay. Imidazole can bind to nickel, and besides that, imidazole can also bind to cobalt. Remember, okay. Then there are two different questions on protein quantification. Try to answer them. At 280 nanometers, a protein solution has an absorbance of A280 is equal to 7. And the molar extinction coefficient, it is given as, I cannot see. Yeah, so E280 it is given as 13460 mole inverse centimeter inverse. So what do you think is the concentration of the protein? So for this you need to apply the Beer-Lambert's law, and you are going to get Beer-Lambert's law questions in your exams. That is with 100% surety. It is going to be there. At least one question on Beer-Lambert's law is going to be there in your exams. So please practice all these questions. Practice the more difficult questions if possible. This is still a very simple question. Okay. So you're saying it's B. What about the next one? Protein absorbs at 280 nanometer due to the presence of which amino acid? There is tyrosine, there is tryptophan, and there is histidine. Yes, it is mostly tryptophan, although. Okay. So let's see. These are the answers. If this would be an MSQ, you would have had to mark both tyrosine and tryptophan. But if this would be a MCQ, you would mark only tryptophan. Okay, because tyrosine actually absorbs at what? 270 around nanometers. Now see, these are the different structures of tryptophan, tyrosine and phenylalanine. All of these are having these conjugate double bond structures. They are having aromatic rings, because of which they are absorbing light at the UV spectrum. Okay, and then regarding this is your Beer-Lambert's law. A is equal to epsilon into C into L, where C is the concentration. So concentration is equal to 7 divided by 13460, and if you do the calculation, you get 0.00052 molar. So that is equal to 0.52 millimolar, right? Now they have given you us the sequence of a protein. Okay, these are the different amino acids of a protein. Now they are asking you how can you know about the molecular weight just by the sequence without doing any experiment? Is it possible to know about the molecular weight, pi, maximum solubility at a specific buffer, extinction coefficient, everything about a protein? They are asking whether you can infer just from the sequence of the protein. Well, it is very much possible. There are bioinformatic tools. Okay, and using these bioinformatic tools, you can learn about different characteristics of a protein. So when you work with DNA, when you work with protein, there are going to be sites which you need to keep in mind, and from there you can know about the different cut sites of proteases in a protein sequence. You will have to know about restriction enzyme sites in a DNA sequence, right? So there is this XPC prot param site which tells you about the different characteristics of a protein only when you give in the sequence of the protein. So let us look at it. See, this is the answer and this is the website. Okay, but how does the website look like? See, so number of amino acid is 76. You have just copied and pasted this entire sequence. So the molecular weight it is giving as 9.5 kilodalton. Theoretical pi is 10 point something, and then the extinction coefficient also they have given, and it is 22460. See, 22460 millimolar per centimeter, right? So everything is given here, and if you look at this is basically the website, okay, for XPC. So this is what comes in when you click on this link, XPC prot param. Then you click on this link, this entire web page opens up. Inside that you give in your sequence of amino acids, and then different parameters they are giving you: amino acid composition, extinction coefficient, everything, theoretical pi, everything they are giving. Okay, so now try to solve this another question. But in the exam, how can we solve it? No, in the exam they will not ask you this question. This is just to tell you about the different bioinformatic tools. Okay. So this is actually a continuation of the previous question. So they are asking you this protein in the previous question is in a buffer of pH 7. Okay, how can it be purified? So see, we understood that the pI of the protein is basically 10.3. So that means if the pH of the buffer is less than the pI of the protein, then what is going to happen? The protein is going to be what? Positively charged, right? So why is it going to be positively charged? Because the NH3 groups, yes. So then you go for cation exchange chromatography, because it is going to be positively charged, because the NH3 groups are going to be NH3 plus, and the COO H group is going to lose this H plus ion. It is going to be COO minus, but again it depends, okay, on the number of CH3 and COO minus and NH3 groups. Now when it is in a buffer of pH less than its pi, the protein is positively charged, and when the pH of the buffer is more than the pi, then the protein is basically negatively charged. So in this case, we would go for cation exchange chromatography, because the protein would be positively charged. Also, you can also go for size exclusion chromatography if you want to purify it. Okay, fine. Now, okay, so we have come to the end of the chapter, and these are all the questions that we had. There are no more questions. So if you have any doubts, you can ask me. Today we completed the class very early, and for the next class, okay, so these are all the different references. So you can have a look at the different references that are here. So there are a lot of different research articles and papers. So if you want to go through them, you can. Okay, I have included all of them here. So If you have any more questions, you can ask me. Can you explain cavitation? I haven't talked about it in this lecture, so I'll have to look into it. I too will have to look into it. What do you want to know about it? I mean, in what respect are you talking? It is there in the MCQs. Cavitation. MCQs of this week. Okay, I will look into it and I will tell you about it in the next class. Okay. Alright, so if there are any more questions, you can ask me. Otherwise, we can end this session today. Thank you for attending the class. Thank you for your cooperation. Thank you so much. You can go to my YouTube channel to get the old lectures, and you can always look up the PPT that I have shared. The entire drive has all the PPTs. You can access that from the NPTEL platform. Thank you so much. So let us end the class now.